Good evening, everyone. My name is Mahmoud Summers, and I'm the host for today's lecture. We thank you for joining us for the 5th lecture of the Oxford Tsarskoe B2 lecture series. For our first-time viewers, this is a weekly online webinar series that hosts a wide variety of keynote speakers from around the world that are at the forefront of COVID-19 research. Our goal for the event series is to invite brilliant minds to give talks that are research-focused and on a wide range of subjects to foster learning, inspiration, and innovation, and provoke conversations that will dictate the future of healthcare. Before we transition to the lecture, I'd like to briefly introduce the organization hosting this event series, upcoming webinars, and our guest speaker for today, Professor Kari Nadeau. In addition, I would like to highlight the procedure in which the viewers may submit questions for the guest speaker, which will be addressed towards the end of the event during a short Q&A session. This event series is hosted and organized by the Oxford University Personalized Medicine Society, a student and faculty-led organization that works to weigh the benefits and detriments of precision healthcare and modern-day medicine. We would like to take this moment to thank our sponsor, the Oxford Center for Personalized Medicine, which is a partnership program between the Wellcome Center for Human Genetics and St. Anne's College, University of Oxford. Throughout the year, we collaborate to hold guest speaker events encompassing a diverse array of subject areas, interdisciplinary symposiums, and field trips for all affiliates of the university. Just this past Tuesday, we held a fantastic webinar entitled Personalized Cancer Medicine, Hope or Hype that you can check out on our YouTube channel, CPM Oxford. If you are a current member of the university and would like to get involved, please register using the above displayed QR code. If you have any questions regarding the society, please email us at oxmed@ox.ac.uk. For today's lecture, Professor Kari Nadeau will be presenting a lecture entitled COVID-19 Immunity in Progress. Professor Nadeau is the Naddisy Foundation Endowed Professor of Medicine and Pediatrics and Director of the Sean M. Parker Center for Allergy and Asthma Research at Stanford University. In addition, she's the Section Chief in Asthma and Allergy in the Pulmonary, Allergy, and Critical Care Division at Stanford and the Senior Director of Clinical Research for the Division of Stanford Medicine Hospitals. She received her MD and PhD from Harvard Medical School, completed her residency in pediatrics at Boston Children's Hospital, and a clinical fellowship in asthma and immunology at Stanford and at the University of California, San Francisco. Throughout her career, she has been a pioneer in the field of translational allergy and immunology, both defining the mechanisms of new therapies and then translating them clinically to make transformative changes in patient care. She has led research in oncology, transplant, and autoimmune trials, and is a member of the National Steering Committee for the intramural clinical research programs at the NIH. She has also started 4 biotech companies in the Bay Area under Stanford patents, and has worked in industry to lead 2 drugs through the FDA to approval. During the COVID-19 pandemic, she led one of the key successful NIH NIAID-funded Remdesivir clinical trials as a potential therapeutic for SARS-CoV 2. Today, Professor Kari Nadeau will be presenting her perspective on the current COVID-19 pandemic. For upcoming webinars pertaining to the lecture series, we have next week, Wednesday, June 10th at 5 p.m. BST, Professor Saad Omer of Yale, and the inaugural director of the Yale Institute of Global Health will be presenting a lecture entitled Consequentialist Research in a Pandemic, the Case of COVID-19. The following week, on Tuesday, June 16th at 5 p.m. BST, Professor Chandan K. Sen of Indiana University and the director of the Indiana Center for Regenerative Medicine and Engineering will be presenting a lecture entitled Electroceutical Management of Pathogens. Recently, his lab group developed an intriguing electroceutical mask that has been shown to neutralize SARS-CoV-2 upon contact. To register for these events, please RSVP using the Eventbrite to ensure your spot for these lectures. For later dates, we have on Friday, June 19th at 5 p.m. British Summer Time, Professor Scott Boyd of Stanford and the lead researcher behind a novel antibody test will be presenting a talk entitled Human Antibody Responses to SARS-CoV-2 Personal and Public. To submit questions for the live Q&A at the conclusion of the event, please use the link/QR code to navigate to a Google form that will allow for you to submit questions. If you'd like to submit multiple questions, please use the form to make multiple submissions. Thank you. And now, without further ado, we'd like to welcome Professor Kari Nadeau to present her talk. Please stand by as we transition to the guest speaker. Thank you so much.
Thank you. Everyone hear me okay?
Yep, thank you.
It's a pleasure to be here today. Thank you for inviting me. My name is Dr. Kari Nadeau, And I'm very excited today to be able to give you a talk on immunity in progress. And I've tried to make sure that we give you the most up-to-date reviews in the literature. So we know that the literature is moving very quickly, and I've tried to make sure that I can reflect its fast pace. But importantly is we only have limited time, so I will try to be able to answer any questions you have as well for anyone that has other information or that we can discuss in terms of what are the implications of some of these findings to date. So I first want to talk about natural immunity, then I'll talk about immunity in therapy, and then immunity in vaccines. This is a recent review in Nature Immunology, and I think this is a very good summary of what might be happening in the human body when someone gets COVID-19. And importantly, we know that through at least 7 different receptors in the human body, the virus co-opts those receptors and uses them as means to get into the cells. And when that occurs, it can cause viral replication, viral-induced lysis, apoptosis, and when that happens, there is a necessary immune response to that virus, which is seen in this screen. There you see that there are natural cytokines being released by the immune system, IL-6, MIP, interferons, and those are mostly mediated by T cells, macrophage, and monocytes. And there are then, after that initial cascade of cytokines and immune reaction to the virus, there's really, in a lot of patients, a bifurcated road. Either they develop neutralizing antibodies, they develop T cell mediated sterilizing immunity, and then they combat the virus. And this oftentimes is associated with more of a mild or asymptomatic disease. But unfortunately, in some patients, we actually see that it leads to many different aspects of vascular permeability. There seems to be some comorbid conditions like insulin resistance that then leads to dysfunctional immune reactivity, which causes major pathology that leads to vasculitis, leads to late onset respiratory disease that can result in death, as well as embolisms and cardiac issues as well. And we're seeing that in different ethnicities, in different genders, this might be more prone than others. And I'm at Stanford now and do work in immunology in the laboratory, but I've also been part of a wonderful team at Stanford looking at these patients in the outpatient setting and inpatient setting. And we have a lot of experience now, but many of our colleagues across the country and across the world are seeing death rates much higher than California. But these central features of the immune system are at play. So I'd like to summarize them. So after an infection, there seems to be a typical 1-week effect, and this involves a self-limiting viral respiratory disease, which ends with the development of a neutralizing antiviral T cell and antibody-mediated immunity. We'll talk about those more specifically today. In addition, there are IgM and IgA and IgG-mediated antibodies. In severe disease, patients show severe lymphopenia, eosinophilia, increased neutrophils. They have extensive pneumonia and lung tissue destruction, but not everyone. Some people actually have more diarrhea. Some people are losing their sense of smell. Some people are getting, as you know, in children, they're getting a specific type of microangiopathy, toxic shock-like syndrome picture. And then in adults, cytokine storms are occurring now, and they can lead to what seems to be an acute respiratory distress syndrome. Lymphopenia seems to cause a defect in antiviral and regulatory immunity. Macrophage and other immune cells release cytokines that can cause catecholamines and amplify the destruction in many tissues. And then importantly, we are also seeing the early detection of acute phase reactants like CRP and D-dimers. I'd like to bring this into the perspective of the patient outcomes, because we know that specifically the immune system is so critical to be able to fight infections, but within that is the very important aspect of antibody production. And so with that, In the mild cases, a lot of times antibodies are produced early in no symptoms. In mild, they're produced slightly later, but importantly is that the contagious period seems to be about 0 to 14 days. However, in mild to moderate symptoms, it seems to be prolonged. Importantly, in severe symptoms, there is a period in all of these cases where there's no symptoms, where people can be contagious. Then people develop severe symptoms in this regard, and then they can get hospitalized, and then antibodies start being produced. And then there is another program in which people are developing critical symptoms. About 5% of people, again, those people can initially present with no symptoms, then critical symptoms there follow therein, and then ICU and ventilation. Unfortunately, this is associated with the highest degree of death. 50% of people so far around the globe, unfortunately, die in this particular category. 15% in this category, and then if someone is mild and stays mild, that's 0, and then 0 here. I think for me as an immunologist, what I really want to understand is the relationship between the T cells, cellular immunity, and the antibodies being produced, and viral shedding over time, so that we can make sure that we can create some kind of biomarker system that we can look at early on during this yellow phase period here, during the no symptoms, so that we can try to predict those people that might go on to develop a more severe scenario and hopefully be able to prevent death with those types of biomarkers available to us. So let's look at immune antibody responses over time. We know, again, like I said, there's this window period that's asymptomatic, that people can still be contagious. There is a period of time where the SARS virus is replicating, as I mentioned before, but over time, after about 14 days, what people have seen now in many studies is that that RNA can decrease, and that is associated with a concomitant increase in IgM and, importantly, IgG. Now, this is a schematic figure, but I'd like to point out the fact that this does not plateau. In some patients, it is decreasing over time, and we need to understand the rate of decay of antibodies to coronavirus. This is similar to other coronaviridae families, but we would presumably, all of us as immunologists as well as the public, would like to make sure that those immunoglobulins can stay high to be able to afford some degree of protection to that individual. And we don't know the long-term consequences of the immunoglobulin increase and then potential decrease over time. So with these different variables of outcomes, there seems to be two main categories that we can look at, and we can potentially differentiate those outcomes with antibody responses. With low-dose SARS infection, i.e. someone might perhaps be in the same room with an individual, and for about 15 minutes they're within 6 feet of that individual, and if they're speaking loudly, it is perhaps the case that they will get a low dose of SARS-CoV-2 infection. Whereas someone that might be within 6 feet away from an individual and spending about 15 minutes with them, and unfortunately that individual coughs in a direct way near the COVID negative individual, they would be presumably receiving a high dose of the virus. And so there are these two potential outcomes given these two scenarios. One in which an effector T and B cell immune response occurs and they develop antibodies that neutralize, and there's rapid clearance and indolent disease. But this does not lead to a severe outcome. There are others in which if they received a high dose of the virus by contact, then they will presumably, with that high viral load, that will induce lymphopenia, immunosuppression, inefficient T and B cells immunity, as well as cytokine storms. And that will result in severe disease. So when we look at this with respect to studies that have occurred, this study was published in China with a total of 173 individuals. And importantly, they examined serially over time 535 samples, and they looked at IgM and IgG to SARS-CoV-2. And they looked at the dynamics of antibodies with respect to more severe versus mild patients. And they looked at seroconversion rates as well. And importantly is the following. When you look at the more severe patients within 1 to 3, 4 to 6, 7 to 9 days, and then importantly 10 to 12 and increase, you can see this curve develop in patients that were more severe compared to those that were less severe. And we think that's because there was an increased antigen exposure and viral load in the severe patients. Now, importantly, many people ask, well, if your antibodies go up, does your virus go down? In the viral secretions, either in tracheal aspirates or in nasal secretions, do you see a concomitant decrease in viral load once the antibodies increase? Because then at least we can infer that there's some functional consequence of the antibodies. And this was published by Liu et al. This is a preprint, and I think I'd want to make sure that all of you see that by about 16 days, the viral detection is decreasing while the antibodies increase. And this is very important. What we need to make sure we understand, though, is what's happening beyond 16 days. If this antibody, perhaps in an extrapolation, decreases over time, what happens to this viral RNA? And these are the studies that need to happen now across the globe for patients that have recovered, and then we need to follow them longitudinally. So when we look at antibody responses, it's very important to look at antibody responses as it relates to the virus and the physiological properties of the virus, both the spike protein as well as other proteins involved in the viral replication and pathology. And it's also important to know whether or not the antibodies are functional, whether or not they neutralize and perhaps inhibit the spike protein from binding to its cognate receptor or binding to any of the receptors in the human body, like the angiotensin converting enzyme receptor. And so here you're seeing that it's important to note that the antibody titers are going up in days after the symptom onset, and you can see the p-value there is highly significant, and that these are associated with neutralization. This was published by Suthar et al. Recently. Endpoint titers are highly variable. Unfortunately, at this current time, we still don't know whether or not this correlates with protection. Specifically, we do know that we can detect them greater than 30 days post-symptom onset, which is helpful. They're developing within 1 to 2 weeks post the symptom onset for the majority of people, although there's some people that it takes much longer. And importantly, is the decay rates over the longer periods of time, as I was mentioning, are not known, and this is something that we need to try to understand. So I've talked about B cells, but what about T cells? This paper was recently published by Alex Setti's group in San Diego by Grafoni et al. It was published in Cell, and it's one of the first papers that we've been able to get an insight into T cell functional responses to the virus. And so what this group did was separate unexposed individuals to COVID-19 exposed individuals. They did an analysis of the immune cell response from recovered patients, and they identified regions of the virus that the immune response targeted, and it actually revealed cross-reactivity with other common circulating coronaviridae. Again, many of these other coronaviridae cause the common cold, and many people have already contracted them in their lifetime. So this just shows you some of the demographics of the individuals. Again, in many of these studies, we don't have a large number of people, but I think it's important that for the preliminary studies that we're doing in COVID, that we can get the necessary cohorts and sample sizes needed to be able to look at differences in the immune system of those that are infected versus those that are not. And so again, they looked at individuals, and most of them in California because this is in San Diego, but I think that you'll see that some of the T cell responses here most likely will represent the global population as well. So what did they see? Well, initially they looked at percentage of cells, both B cells and T cells and monocytes and other cells as well. And you can see in the COVID positive patients versus those that are negative, there's an increase in CD4s and this trend for a decrease in CD8s, although not significant. And then importantly, they did see a decrease in B cells, CD19 positive cells. And what I think really is remarkable about this paper is they took pools of SARS-CoV-2 derived peptides. And that's important. So when we look at some of the non-structural proteins, the open reading frames 1A and 1B, and again, this is a very large protein, larger than dengue, larger than other viridae. So within that, there are a lot of antigenic determinants that could be associated with immune reactions. And S stands for spike protein. When we look at the RNA and translation in a human cell, there are many different peptides that are expressed, accounting for 7 common HLA-DR alleles. 221 is what they tested. The spike protein CD4 positive pool of 15 more overlapping peptides were tested as well. This was 253. They also looked at CD8 epitopes, accounting for 12 common HLA-A and HLA-B alleles, split into pools A and B, and this were 314 peptides each. And then they incubated with 1 million PBMCs for 24 hours to look at activation profiles. And with that, I want to show you first the data from the CD4 positive T cell population. This was, again, the stimulation of the megapool of peptides derived from either the spike protein or all other non-spike proteins associated with the virus. And you see this incredible increase of CD4 positive T cells with OX40 positive and CD137 positive expression. And this is just an example of how they gated those cells. And you'd want to look at the increase in a particular cell, but you also want to look functionally as what is that cell secreting. And what we found interesting in this is that that T cell population, the CD4 positive T cell positive population, was expressing IL-2 to the same degree, relatively speaking, to non-spike protein peptides as spike protein peptides. So again, it's important that perhaps in vaccines, that the whole virus is expressed in a human to be able to get at both of these features of the viral immune reaction in our T cells. This was IL-2 expression. This is interferon gamma expression. So we see that interferon gamma is an important mediator, most likely, in being able to combat the virus, both non-spike protein moieties as well as spike protein moieties. So now let's look at the CD8 population. You can see here that CD8s are also relatively increased in the peptide pools that they use. There's a lot more variability than the CD4 population. And what's interesting is that CD8s, of course, as we know, are critical to cytotoxic T cell responses when fighting viruses. So it's important to see that there were increases here. In addition, this can be shown in terms of relative differences between how CD8s responded to, for example, CMV versus COVID-19. And again, there seems to be an increased reactivity of the CD8 cells to both COVID and CMV. And again, when we look at the mediators of potential CD8 function, granzyme B was increased, TNF alpha, again, increased in COVID, and IL-10 was downregulated or decreased in the interferon gamma secreting CD8 T cells. I thought this was also interesting, and one thing that also came out in this paper was that in the patients that had prior infections to coronavirus, either the beta coronaviridae or the alpha coronavirus, that it was important to note they did have antibodies to those, but not to SARS. So now let's look at these peptide pools and looking at the frequency and abundance of these proteins. What's clear is that those proteins that are most abundant, those proteins that probably had the most antigenic exposure via dendritic cells and antigen presenting cells, you can see that that was associated with the highest amount of CD4 T cell stimulation index compared to other peptides within these regions. Again, much higher in the open reading frame part of the protein, even compared to the spike protein. So it's important to note that the entire virus is eliciting a response of the immune system in the T cells, as well as most likely the B cells. So in conclusion, measuring immunity to SARS is key for understanding COVID-19. It's important to develop peptide pools and look at both CD4 and CD8 cell responses. and that T cell responses are focused not only on spike, but also on many of the other proteins, and that the T cell reactivity of SARS epitopes is also detected in non-exposed individuals as well, meaning that most likely the other coronaviridae family of viruses is potentially allowing people to recognize the SARS epitopes when they do get a SARS-CoV-2 infection. So again, another brief summary via a pictorial figure that was recently published. And you can see overall in patients that have been infected with COVID, there's a decrease in peripheral blood T cells. There's an increased activated CD4 and CD8 population. However, there's exhaustion over time in these phenotypes. And importantly, in severe disease, there are specific inflammatory mediators that the CD4 positive T cells are responding to. So peripheral blood T cell counts and viral load seem to be negatively correlated with each other. So now let's move on. So I've talked about B cells and T cells and our immunity. But what about natural killer cells, monocytes, other populations? This was a paper that was recently published in Nature Immunology, where single cell analysis and single cell sorting with RNA-seq actually profiled patients who were hospitalized with COVID, and they looked across the monocyte and natural killer populations. And what was, I think, for many of the scientists' interests, they were profoundly interested in the fact that the NK cell populations, the dendritic cell populations, were significantly depleted in patients with severe disease. So you can see here in this UMAP of the different cell populations and clusters in healthy versus COVID patients, the COVID patients being in orange and red. You can see the dramatic differences in these populations. If you map them over where we would expect to see these cell clusters and cell phenotypes, you can see there's a profound difference in this population and then also in the T cell populations. Finally, what does the immune system look like in someone that has recovered? And again, those that were recovering versus controls had more B cells and NK cells and T cells, less myeloid cells. And it's important to also notice that even the recovering patients had decreased lymphocytes and increased myeloid cells, and inflammatory cytokines like IL-1 beta, CCL3, IRF1, JUN, and FOS all were expressed at high levels. And it's important to be able to look at single cell omics like this particular paper published here by Wen et al. In Cell Discovery, so that we can really get a feeling for what immune profile might be able to protect someone long-term from reinfection as well. So with that, I've been talking a lot about what's known so far in many different cell subsets. The NIH has recently funded a study in which they would like to look at patients long-term. And this is happening at Stanford as well as other sites around the US. And the primary objective here is to look at immunological assessments and severity of illness in all phenotypes of the immune system, and look at concurrent participation in many clinical trials that are going on around the country, as well as off-label use of some investigational drugs, to see to what extent the immune system changes with these particular agents. And we want to also look at genetics, clinical outcomes, immunological variables, and this is a large study going on across the US. And you can see here that we'll be profiling patients from hospital admission while they're in the hospital, some of them more severe than others, and then looking at the recovery phase from 3, 6, 9, and 12 months. So we're excited to look at this, and this is going on with Mark Davis, Bali Palandran, Holden Maker in our Stanford site, as well as many other sites around the country. So I'd like to quickly go through immunity and therapy. We know that we can understand how important a specific immune cell is or a pathway is, depending upon how someone potentially responds to therapy. And in the era of personalized medicine, we need to be able to look at a panoply of candidates that could perhaps affect the immune system. And there are many out there now being tried in COVID patients, more than this list probably allows. But Camostat, for example, is a protease inhibitor, Hydroorotate dehydrogenase inhibitors also affect pyrimidine synthesis and most likely will affect T cell pathways as well as inhibit viruses directly. JAK-STAT inhibitors are being used right now. Monoclonal antibodies to the spike protein and other proteins are now being used in the clinic. And anti-GM-CSF, anti-IL-6 receptor antibody, interferon-1 beta, and then convalescent plasma. I'll talk quickly about some of these now. Interferon-1 beta is importantly being used in clinical trials. Could it perhaps have a role in prevention or a role in very early mild disease so that one does not then go on to develop severe disease? And this was an original study that was recently published showing that indeed viral load was significantly different and the rate of decay of viral load was significant in the patients that were treated compared to those that were not treated with interferon-1 beta. Another series of antibodies have now been moving forward in clinical development by many companies, and the IL-6 receptor blockade is important for those late stage, highly inflammatory reactions that are happening for patients that are severe and in the hospital with an ARDS-like picture, an acute respiratory distress syndrome-like picture, as well as pneumonia. Finally, convalescent plasma, I think, is picking up a lot of speed. I show this picture of the US, but of course other countries as well are looking at convalescent plasma as a potential therapeutic moiety, and this is the only polyclonal antibody-based therapy we have available at the time. However, we need to be careful and cautious and show equipoise in analyzing any of these therapies. For example, with convalescent plasma, we're still trying to figure out the exact safety. There have been some transfusion reactions, but importantly is the fact that from a pilot proof of concept that by infusing others polyclonal antibodies into patients, it seems to be protective to some degree, again, showing that those antibodies could potentially be neutralizing. So now moving forward with, I think, the topic that is on everyone's mind, and that is immunity and vaccines. This is very important. Obviously, we'd like to be able to have a vaccine for the globe, and what is that going to entail? There are key questions in vaccine development. This slide was made by my colleague Bali Palandran, and these are critical questions that we're all asking as scientists. Do infected people develop potent and durable neutralizing antibodies? Does infection protect you from future infection? How long does immunity last? What are the correlates? What kind of immune response do you need? Is it just antibodies or T cell as well? Most likely both. How can a vaccine stimulate robust and durable antibody responses? What roles do T cells play? Is there a risk of antibody-mediated enhancement? And then more than 100 vaccines are being developed. Which ones will move forward? So this was a great study that was published by Bali and his laboratory. And it's important to note that even though this was a yellow fever vaccine, this has applications for any kind of assay that we need to look long-term about whether or not someone is responding to COVID vaccine. We need to look at antibodies, as I've shown you today, but also look at T cell antibody responses in response to each other and being able to find out what other cells are involved, NK, dendritic cells as well. Catherine Blish in our group is looking at NK responses. So I think many immunologists will come to the table and try to understand how these vaccines are working. Finally, global access. How do we vaccinate all the people in the world? There's a timeline that typically takes about 5 years. We're trying to crunch that into 18 months now around the globe. How is that going to be possible? We need to be rigorous and test this carefully and be cautious, but also efficient and effective in bringing this vaccine out. There are many challenges in terms of stability and making sure this is a product that's thermostable. We need to make sure that we get proper doses for vaccination, again, making sure that immune response is optimal. And then, of course, there are many other issues ethically in terms of distribution, making sure it's fair and equitable to the populations that need it most, and to be able to understand how age is affected by vaccination in the immune system. Mark Davis in our group and Jorg Granzi has done a lot of work on this. There's different vaccine types, DNA vaccine-based vaccines, RNA-based vaccines. I understand that you've been taught by other people here at the Oxford series about these other vaccines that are currently being used. There's non-replicating viral vectors, inactivated virus, and live attenuated vaccines. And of the vaccines that have been used right now in the exploratory phase, there are many that are moving forward, but very few in phase 1 right now. So I'd like to point out that of the different types of categories of vaccines, there are 2 that seem to be moving quickly and have some results so far, as well as the University of Oxford vaccine that is being cooperatively clinically developed with AstraZeneca, and that had positive safety results. So again, very few patients right now that we have some immune responses, T-cell and antibody responses to these types of viral vectors, and we want to make sure that we follow these over time. And how do we look at increasing the capacity around the world? How do we decrease timelines? There's an NIH accelerator now that is moving forward thanks to Francis Collins and others and the FDA. The Emergency Use Authorization Act of the FDA is allowing us to decrease timelines, but in that the FDA is still going to be very rigorous in its assessments, as I'm sure the global regulatory agencies are as well. We have developed this warp speed program here in the US, but that's not necessarily new. That means that there'll be mass production of vaccines and then testing after that. And that happens similarly in the polio era with the Salk vaccine. The best and safest of the vaccines are not necessarily those that came out first, and so we need to be careful and test these. And then when we look at a trial and how fast these vaccines will be available to the world, we're looking at a trial that's most likely 20,000 active versus about 10,000 placebo through BARDA and funding. This hopefully will be happening quickly and starting in July. People have predicted that it's possible in the best case scenario that we could have a vaccine available in the fall, or potentially next fall as well. We need to really think about these timelines and not make any promises, and make sure that we increase the public trust in vaccinations and be careful about when we promise this vaccine and when it does come out, to make sure that it's equitably distributed and assayed for immune profiling. And I want to thank everyone for their time, and I'll take questions. Thank you.
Thank you, Professor Kari Nadeau, for that wonderful lecture. And now we'll transition into the live Q&A. So throughout the event, we've had multiple questions submitted. So let's just go through first one. Okay, so for the first question, it says, I've heard that even if you have antibodies for the virus, it doesn't mean that you are immune. How do you go about proving/disproving this?
Right, great. So there are many people studying this now. For the people that are B-cell biologists and looking at this, like Scott Boyd, who will be talking subsequently in June in one of your lectures, he's developing an assay where instead of having to virally replicate in situ, and that needs a higher level type laboratory facility and licensure, instead of growing the virus in culture, what you can look at is to what degree the antibodies block the spike protein from binding to its cognate receptor. And so with that, what he's showing so far, and he's best at being able to eloquently talk about his assay as well as other assays, is that by using patients' plasma and patients' antibodies, he's able to show that he's able to inhibit the spike protein of the virus from binding to the receptor. And he can infer, based on those patients and based on how well they've done, that that actually is a neutralizing antibody. Other people around the world are developing other types of assays, but that's one example of being able to get at function as well as quantity, and looking at stoichiometry of how much of the polyclonal antibody and how many epitopes do you actually have to bind to inhibit the virus from replicating and from being able to undergo antibody-dependent cell killing.
Thank you. The next question submitted says that, what is the role of neutrophils in disease severity? Are they friends or foes?
Great question. There's a lot of data now on IL-17 and activation of T cells and IL-17 profiles, which of course is a growth factor for neutrophils. Originally, some of the data coming out from China showed that the ratio of lymphocytes to neutrophils was a harbinger for severe disease. So we know that relative lymphopenia, increased neutrophils, somehow is a biomarker for severe disease. I believe we have neutrophils for a reason. They're our first responders. We make many neutrophils in any given day. Their half-life is about 6 hours, so we need them to fight infections. But oftentimes, if that reaction has been too exuberant, oftentimes the neutrophil mediators can actually cause tissue breakdown and can cause problems with tissue inflammation. I think that's what happens. If you have neutrophils in the beginning, that's great. You can try to fight the virus, but unfortunately, if the neutrophils stay high for a long period of time, then we create some of the tissue damage in the host response.
Okay, thank you. And for the next question, would the potential biomarkers that will be produced imitate the structure and nature of an ACE2 receptor, or would it have a mobile function with an unspecified positioning in the bloodstream?
That's a great question. I actually don't know the exact answer to that. I think we'll have to see. I do know that different populations have different expressions of the ACE receptor, and that seems to also play a role in whether or not they get severe disease or not in terms of their viral load and how much the virus affects them. But that's a question that I would defer to others.
Thank you. And for the next question, it appears that incorporating both spike and non-spike proteins in a vaccine may be beneficial for protection. How would this affect the way vaccines are produced, and would it pose additional difficulties? And to what extent would an attenuated form of SARS-CoV B2 address this issue?
Right. Most of the viruses and vaccines that we use right now, many of you got vaccinated, hopefully, are from live attenuated or killed viruses. And so with that, you get the full display of antigen determinants that are needed for a proper immune reaction. But even that, obviously, not all people have a full response even to live attenuated or to heat-killed vaccines. I think to the extent that we use recombinant technology now to be able to generate vaccines, with some of the RNA vaccines coming out, there's a lot of issues around RNA stability. I think we have to be careful and use our science to make sure that we can modify any recombinant vaccine and put in the proper RNA and the DNA encoded regions so that we can get a good display of the key antigens. And it might not be the entire virus. To what degree do you need a certain amount of antibodies and a T-cell response to be able to quench the virus? If we can at least get it to 70%, is that the threshold we need to be able to then have an effective outcome? That's hard to know. So right now we think that having all the proteins is important, as what was shown in the Grafoni et al. Paper. But exactly how much, what the threshold is, we don't know. So I think we'll have to see as these other viral type categories in vaccine research move forward.
Thank you. And for the next question, transfer of plasma from recovered patients to patients suffering from COVID-19 has shown variability in effectiveness. What could explain this variability?
Yeah, I think convalescent plasma use, we're still trying to figure out the best titers and trying to figure out what the best antigen determinants were that these antibodies were made to. And the typical convalescent plasma, we're really not looking at all of those factors before we give the convalescent plasma to the recipient. So there are a lot of variables one needs to look at. Antibodies that one is giving to a recipient, and that could be somewhat quantitative, but also the function of those antibodies. So I think that's why we're seeing different responses, but that comes from the donor. There are other variables in terms of the recipient themselves that also play a role in how well they're going to improve, and that pertains to comorbidities like obesity, diabetes, gender. There are many issues now, and age, that might affect how someone responds to convalescent plasma.
Okay, thank you. And for the next question, has there been any estimates of how long beta and T memory cells provide protection from the virus in an individual who has fully recovered?
Excellent question. Exactly what we need to study, and that's why this study called IMPACT, and hopefully many other immunologists around the country are making sure they can follow the patients that have recovered over time. We're doing the study here at Stanford where we're studying people over a year to be able to best assess, as well as their household contacts, to what extent are they shedding virus, to what extent are they now immune, and then to what extent, if there's another surge of the virus, will they be protected against reinfection. And so this is critical for understanding if there is a specific signature of T cell and B cell responses which allows someone to sustain and have sterilization in their immunity, that would be great. We can get that type of signature, that hopefully will help in our vaccine researchers as well as people doing therapy in COVID to be able to try to get to that signature that allows for and provides for sustained immunity to any virus with COVID-19.
Okay, thank you. And for the next question, do you think the immunity that has been developed due to encounters with previous members of coronaviruses affects how our body fights against SARS-CoV-2?
That's a great question. People are looking into that as well. You know, one of the thoughts is that children are relatively protected because they've seen more frequently many of the other cousins of the coronaviridae family, i.e. the ones that cause the common cold. I don't know if that's true yet. We actually need to study that via rigorous immune testing. From the Grifoni et al. Paper, you would infer that it is possible that if someone has recently seen a coronavirus cousin, alpha or beta, that would potentially protect you when you're going to be exposed to SARS-CoV-2, but it's not perfect. So it's not as if we should go run around and try to get these other coronaviridae infections to try to protect us against COVID-19. That didn't necessarily always happen.
Thank you. And for the next question, is there a known biological mechanism for the lymphocytopenia seen in severe COVID-19? Which viral and host factors may be interacting?
Yeah, great question. So, you know, a lot of immunologists are looking at whether or not the virus itself replicates in immune cells. We know that it replicates in some endothelium and epithelium, but does it actually replicate immune cells? If it does, that could be one direct way that it's causing killing, right? Just like kind of HIV. But right now, if you were to look at that, you would, because it's a positive strand RNA virus, you would look at whether or not you can detect negative RNA strands of the same strand in the immune cell to see if it was replicating in the immune cell and causing lymphopenia and cell death. Right now, there's no one that I know of, at least. Feel free if those people on the line have otherwise found differently. But I think that right now, we do not think it's via direct viral replication in the immune cells, that it's viral mediated lysis through granzyme B or through perforin pathways and through receptors like CD147, where the virus gets into the cell and can cause cell death. So right now, that's what we think is happening, although we don't know all the etiology.
Okay, thank you. And for the final question, is SARS-CoV-2 capable of downregulating MHC class 1 molecules as an immune evasion strategy?
Oh, no, that's a great question. I don't know the answer to that. I wonder if there's something in literature already. There should be something in literature. We should look into that. Thank you.
Okay, and I think that covers the last question submitted. Thank you, Professor Kari Nadeau, for an excellent talk and for taking the time to answer some of the most pressing questions that our audience had concerning the immunology of COVID-19. In addition, thank you to our audience for attending the webinar live, and we hope you enjoyed the event. This lecture and Q&A will be shared on our university website and associated YouTube channel as soon as possible. Next week, we have a lecture to be presented by Professor Saad Omer of Yale, so please make sure to RSVP for that event. Thank you once again, Professor Kari Nadeau, for presenting such a wonderful lecture, and we hope to see you all for next week's lecture.
Thank you very much. Good luck, everyone. Get a lot of research done. Thank you.
Thank you.